Invitation. First slides are about the Biomarkers of Aging Consortium. So I would really like to encourage you to look at their website, their conferences, their prizes. I was also happy and grateful to be part of 2 of their papers, generally on biomarkers of aging, also on the important aspect of validating biomarkers of aging. Just in brief, biomarkers of aging, where you want to predict future health and survival. And if you've got better values, you assume you've got longer health and survival. And the other use is, of course, for predicting suitable interventions. So you want to match your biomarker values to intervention data, where people with similar values benefited from that intervention. And that could be a good hint that the intervention is good for you. Regarding interventions, one of them is a nutraceutical, quercetin. And some people take 500 milligrams of quercetin every day. And it's a question, does that match their biomarkers? And very simple biomarkers are just listed there. And asking AI, this was already done a few months ago. This was Claude. You get kind of okay recommendations. So it's assessing the biomarkers in relation to quercetin. The question is now, what is how do you measure the quality of any such advice? And just generally, of course, you're expecting correctness. That's maybe the most important. So you want to use benchmark tools. You want to also check your input data quality. Essentially, you want to check everything. Correctness is maybe the most important. But there's also usefulness and comprehensiveness. And there AI is pretty good. Large language models, maybe with retrieval augmented generation, maybe using knowledge graphs, that can make a difference. There are models for meta-analysis. And what's not so easy is interpretability. But there's also progress there in explainable AI. And now going more to intervention advice, what you want is you want to consider causality and use data from randomized controlled trials. For example, you want to consider side effects, toxicology, maybe look at adverse outcome pathways from toxicology, research, and application. You want to consider more interdisciplinary evidence. That's next point, social factors, feasibility, sustainability. And maybe also for reporting and analysis, you want to consider some standards. And finally, for longevity, which is a topic here, specifically, I think you want to consider large-scale human longitudinal data, like UK Biobank, NHANES, and you want to consider known anti-aging or aging-associated processes, like the hallmarks of aging, pillars of aging. And if you've got other ideas, just get in touch with me. I'm interested in completing this list. Now back to this question about quercetin. What you can now do, very simple, you can summarize those requirements, and you can add it to the prompt. In this case, it's actually our best example, we really get a much better result, because a large language model is now saying that there's a concern with quercetin, which is interacting with atrial fibrillation. And atrial fibrillation was actually mentioned as one of the clinical biomarkers. And they're competing for the same enzyme, for example, quercetin and warfarin. So this is like, Jackie also had an example which is kind of insightful. So you've got quite some surprising insight here, but it's also our best example so far. And Anton, who's also here in the audience, did actually a systematic analysis about taking rapamycin. So what you see here is in columns, you've got all those various requirements, like correctness, usefulness, and so on. And then in columns, you've got the output of Claude without the requirements and with requirements, all marked by the quality. So this is good, very good, this is okay, and if you've got an X, that was a bad response. And the assessment was actually done by another model. So here Claude without requirement and with requirement is assessed by GPT, and here GPT without requirements and with requirements is assessed by Claude. And you can see, especially by GPT, that Claude thinks the results are much better if you're including those requirements. And also, especially for this holistic theme of also toxicity and side effects, you get better results actually for Claude and for GPT. This is still work in progress, specifically systematic benchmarking studies. We have got a perspective submitted, and it's also hopefully soon appearing in arXiv. As I said, Anton is here, and I'm also really happy to talk to you about those topics. And to summarize this part, so essentially I said, what are the criteria? I suggested that especially retrieval augmented generation and knowledge graphs would be an important amendment. Visualization is another issue for interpretability and explainability. And we are now trying it out in Rostock in the Institute also to generate health and productivity advisory tools, to do intervention analytics based on omics data, and also we've got a strong interest in cancer molecular tumor boards. Now I'm coming to the second part. Now I would like to show you a little bit how AI could be used to design the ultimate longevity study. This is done with GPT-4, actually via Perplexity, and it's a dialogue. So first of all, setting up which are the important considerations, and then executing those considerations. Like PQ is a standard scheme for population, intervention, comparator, and outcome. That's a standard for study designs. But also important is feasibility, for example, maximum return of insights, of course. And I made it openly available, the dialogue. And this is a start. So I said it's an incomplete list. Essentially, large language models are very nice for the short tail of knowledge, where there's lots of frequent statements. So immediately you get a reference to ethical considerations. Sorry. Yeah, ethical considerations. But you also get something about comprehensive data collection and adaptive study design, which is also important ideas here. And then if you ask it for really executing the design, showing you something, you're actually getting pretty boring results. That's because it's a short tail. Things are summarized that are very frequent in the training data. Like you get a pretty boring dietary intervention study design, an exercise intervention study design, and maybe a bit more interesting design on a pharmacology intervention study. But you can, of course, try to nudge those models into becoming more innovative. And this is the next step here. So it's actually a pretty good personalized nutritional genomics study here. And microbiome modulation study, study number 2. And senolytic drug intervention study, study number 3, that is designed. And yeah, it says, okay, there's a novel senolytic drug, dasatinib and quercetin. It's probably not that novel. So maybe we nudge it even further. We ask for even more innovative designs. And then you get this design, which I kind of like. It's a partial epigenetic reprogramming study. It's in 3 arms. So the first arm is receiving epigenetic modulators like DNMT inhibitor, histone deacetylase inhibitor. It's also giving some good examples. The second arm is about a chemical cocktail designed to mimic the effects of reprogramming factors. It then actually refers to the 2C and I think 7C cocktails published recently. And the third arm is then a combination of this epigenetic intervention with NAD precursors. And there you get a nice reference actually to a paper where NAD is actually useful to enhance reprogramming efficiency in mesenchymal stem cells. Yes, and you can go further, but then you really get more and more hallucinations that are not very useful. Essentially, you wanted to hallucinate something if you want to have an innovative design, but you also don't want to have too much creativity here. And this balance is probably an important point to consider and to research further. Then, but boring inclusion and exclusion criteria. This is a short tail again, pretty nice summarizing of frequent statements. What it pretty much fails on is to really do a power analysis. What's also failing is the cost. Not in the standard setup, it's not able to calculate the costs reasonably well. Yeah, and summarizing again into slides, for example, is very nice. And here, what I did is actually I added those requirements from the beginning. correctness, usefulness, and so on. And you indeed get a better design to some degree. So if you take a look, also it's talking about causality now and showing statistical power, longitudinal study design. But the point is actually, if you look closer, it's just name dropping. It mentions those terms, but it does not really, in this case, think much about it. And we are working on getting also better there. This is by GPT, so Claude was asked to judge it, and it was very happy, 6 times A, 1 times B now. And if you go next here, that was the old output. Claude is not so happy with that old output. But here we really need to do systematic studies. And there's people working on those tools in general, but I don't know who's really doing it for longevity and aging research. And what I really would like to suggest is to have a leaderboard to find this ultimate longevity study design based on criteria that should be set in a maximally objective manner. And next is now going for mouse. For mice, it's important which kind of mice you want to take. And I found it interesting that after some nudging, actually the F1 hybrids are suggested to be the mice of choice, also for reproducibility. And this is in a major project, also together with Mats. This is now the idea we are pursuing. Also it's listing some pros, some cons, for example. And I said briefly, I'm interested in health and productivity. Of course, I also asked it for this, because health goes with productivity and vice versa. So this is now evolving into a study design for health and productivity. And it's all as adults at risk of cognitive decline as a population for the study. Intervention arm 2 is quite interesting, combining polyphenols with probiotics, which is definitely a good idea. And we're also looking at that further. But some other ideas like Rhodelia, Bacopa, for example, those are substances from Ayurveda who are said to improve cognition and be also for longevity. That's missing in this case still. Regarding benchmarking, we are now closely collaborating with the Biochatter people, Sebastian Lobentanzer and others. This is, we are now members of the consortium, also thanks to Anton. And Anton is also presenting 2 posters here, one on longevity genie, where he is actually implying the Biochatter framework, and it's longevity advised by AI and LLMs. So please check out the posters. He's also doing that with Nikolai Uzanov and others, and also on Just DNA Sec, longevity genetics with large language models. And finally, I'm looking for business-to-business customers, actually. We are starting our own company in Rostock. We have ministry funding. We are looking for pilot customers, cooperation partners, using AI and classical bioinformatics. And we want to do intervention analysis, repurposing, also with a focus on safety and side effects. And any intervention goes. We invest in partial in vivo reprogramming in nutrition, which just needs a good biomarker data. Then I'm coming to the end. I'm welcoming you to Rostock, actually, which is 5 hours away if you take the train and the bus and the ferry. And it's a country of long and healthy life, we suggest. And we will have a longevity week or longevity day end of October. And I thank you all the team members and the funders. I thank you for your attention.
Thank you. Got time for 3-4 questions.
Hi, thank you very much. Very interesting talk. I find this very, very promising, the use of the LLMs for science in general. One question I have, you might have addressed it, but you don't think you did. At the end of the day, the LLMs are just as good as the data you have and the information that it can access. But at the same time, one of the concerns I have is that there is a lot out there that it's either exaggerated claims that are not backed up by what it's in there. There are caveats. For example, I have seen that some stuff in autophagy works very nicely in certain cell types. For example, it doesn't work at all in other cell types. How can all these problems be, gone through to get something useful?
I have actually searched for half an hour, and I have not found any research on the. Maybe I looked wrong, and also the AI didn't find anything. Because I think it's really important to essentially the sweet spot of some hallucination, some creativity, some innovativeness, but not too much. And also then the LLM should, of course, consider all the data that is available, so that you hope that you get kind of, meta-analytic, clearly backed up suggestions. That sweet spot is really the problem. And systematic research on that is probably really important, that you get good designs, but no nonsense designs.
Really interesting talk, right here.
Sorry,
in
where?
the middle.
Okay.
Raghav, right here.
Yes.
So my question is that you showed a lot of very interesting qualitative validation that could be very interesting. It's really interesting that you showed that. My question is that what about a little more sort of quantitative validation of your models, right? You talked about sort of validating with this biochatter company. I'm forgetting. Sorry, I apologize if I'm messing up that name. But could you talk a little more about what that validation could look like, and specifically in this context of sort of longevity interventions and longevity models? Thank you.
Yeah, if you want to benchmark those models, so one possibility is to actually try to come up with a data set yourself, which was created by human experts. And actually, Anton, just 1 second. And so a human expert is one idea, but you can also use AI tools to create benchmark data sets, but then you have to maybe check them yourself, of course, and you always have to worry that there may be contamination, that the benchmark data part of it are already in the training data set. But Anton is much more involved into that, so please, yes.
So what I want to say is there are several ways, because depending on the task, there are things that are easier to validate and quantitate. Well, actually, most of the benchmark, if you look at most of the benchmarks that people use for LLMs, it's mostly like multi-choice questions, or it's some kind of cases when you have ground truths and you can have Rouge metrics or something similar. So such things, in terms of, for example, when you have you want to check whether the model is using some data from knowledge graphs, you actually have check that the data is there, you ask it to generate a structure, you check that some terms are there. So these same things, they are being done. If you look into biochatter code, you can actually see some of the tests like this. With things that are more qualitative in nature, for example, like suggesting evaluating a study, it's way harder because you cannot just have multi-choice question, because it can be done in so many ways. And there we don't have a real ground truth that we can compare how far it is from. So there we have some indirect metrics. Obviously, one of the things that you can do always is make a panel of humans, but it will be too expensive. So what we really want, and I think what can move the field forward, is we have maybe not as good as humans by evaluations, but some automatic benchmarks that can give some hints. For example, the simple thing that Georg showed is when we have estimations by multiple models, it also gives hints like what is wrong, what is bad. Also, when you have additional step in agentic pipelines, like reflection, when the model goes through the answer and checks all the data if it is exactly the true. It's actually showed the role in, I believe, BioCipher papers. There was actually a proof that it improves the input and output, and that it was output of the LLMs. And also there are comparative ways when, for example, you can compare answers. Well, I actually saw this here when you compare several answers and you use LLM-based judge to say what was better and what was worse. It's similar to LLM arenas type, but unfortunately, you have these indirect evaluations, but I don't think we will have in the nearest term really good quantitative scores, because it's too complex to evaluate even for humans.
Great. If you also have more questions, you can ask Anton. No, one more question from the gentleman.
Sorry, just a quick one. How do you filter good knowledge versus bad knowledge? And have you tried to use Cite and Consensus along those lines?
Have I tried to use?
Cite.ai and Consensus.app. They interactive papers.
Not in particular those sites. And one idea that Anton also mentions is knowledge graphs. So knowledge graphs, they can represent knowledge that you then deem to be the correct knowledge. But it's also a very, very difficult task to
I think it was
delineate
just
that.
checking like consensus as and so there are like several consensus methods when
Anton, 30 seconds.
when you like get estimations by several models if they agree or not and discard where there are disagreements. So this we also sometimes tried. And the second, what Georg said, is that you can generate some formal structure like a graph that you can validate with other methods. That makes validation easier because with text you can make it in so many different ways.
Perfect. Great. Thank you.
Okay.